everyone. In this video, we're going to look at the structure and function of the cytoskeleton and the external structures of both plant and animal cells. There are three main components to the cytoskeleton in eukaryotic cells, and they vary in size, location, and function. These images show each type of fiber stained in a different color. Microtubules, which are the largest, in green, intermediate filaments in yellow, and microfilaments, which are the smallest, in red. As the name suggests, microtubules are tube structures and are assembled from dimers of a protein called tubulin, wrapped in a helical formation to form a hollow tube approximately 25 nanometers in diameter. These are dynamic structures within the cell and serve as the cell's highway network, allowing motor proteins to transport various cargo, from vesicles to organelles and even chromosomes during cell division, by effectively walking along the microtubule tracks using ATP hydrolysis to provide the necessary energy. As the name suggests, intermediate filaments are in the middle when it comes to size, typically in the range of 8 to 12 nanometers in diameter. Rather than the tube structure found in microtubules, intermediate filaments are more rope-like and remain relatively static within the cell in contrast to the other two types, which change more frequently. Intermediate filaments predominantly serve a structural support function within the cell, helping to maintain the position of organelles and forming adhesions to attach the cell to its neighbors. The smallest of the cytoskeletal components are the microfilaments, composed of two twisted strands of actin polymers. Like microtubules, microfilaments predominantly assist with motion. Rearrangement of actin filaments under the plasma membrane can create amoeboid-type movement, allowing the whole cell to move. And microfilaments are also involved in circulating the cytoplasm throughout the cell in a process called cytoplasmic streaming. Additionally, microfilaments have an important role in contractility, either in combination with other proteins to allow for muscle contraction, or in separating newly formed daughter cells during cytokinesis. We mentioned the role of intermediate filaments in cell adhesions, so let's have a closer look at three of the main types of junctions between animal cells. We'll start near the top of these cells with a structure called a tight junction. These are commonly found in epithelial tissue, where they link neighboring cells together and separate compartments on either side of the cell. If you consider a scenario like your small intestine, you have digested food on one side of the cells and extracellular fluid on the other. To make sure that waste products and unwanted substances don't make it into the bloodstream, we need to have a system that separates what's in the intestine from what's in the rest of the body, and tight junctions are an important part of that system. Moving down to the middle of the cell, we have desmosomes. These are the junctions attached to the intermediate filaments, and their job is to anchor the cells to each other in a tissue. The third type of junction that we're going to look at are the gap junctions. These are dynamic channels that can close and open to allow the passage of small molecules between neighboring cells, allowing for increased cellular communication. Outside of animal cells, we have a network of protein fibers called the extracellular matrix. It's important to note that this isn't found in plant cells, as they have their own extracellular structures, which we'll look at in a moment. The extracellular matrix, or ECM, has several important roles in animals, including maintaining tissue integrity and cell attachment, communication with cells about their external environment through proteins like integrins, shown here in blue, which help to relay information through the plasma membrane to intracellular signaling proteins, and the regulation of cell growth by storing or releasing growth factors to control rates of cell division. The cell wall is the external structure surrounding plant cells. It's composed predominantly of cellulose, polymer of glucose hydrogen bonded into rope-like fibers to provide structural support, protection, and maintain water balance by limiting the volume of the cell. Similar to gap junctions in animal cells, plant cell walls contain channels called plasmodesmata. which allow passage of materials and communication between neighboring cells. Thanks for watching, and please make sure to check out the other videos in this section.